Good morning, everyone, and I too would like to extend my thanks to NIST for the opportunity to be here and interact with both NIST researchers as well as all of you who have taken time out to be part of this today. So what I'd like to do is just spend a few minutes kind of walking us through how we got to where we are, and really what we're looking for as an outcome in relation to today's activities. So as Marie pointed out, we are the Gut Microbiome Committee with the goal to further understanding of the relationship between food, the human gut microbiome, and its impact on health. And it's really its impact on health which is a critical piece of where our discussions in this area lie. Long-term goal to build a framework for substantiating the benefits and safety of dietary components for establishing and maintaining a healthy gut microbiome. The wording of this goal is sort of shifting a bit, I think, as we come to recognize the complexities of trying to define a healthy gut microbiome and what does that mean in the context of health in general, and where does this fit? So it may be more that a healthy gut microbiome shifts to more the overall impact of the microbiome on health in general. So I think certainly up for debate there. So just a premise, and a lot of this comes out of the food industry side of the ILSI discussions. However, it also is highly relevant in relation to academic research too. The idea that validated indicators for which a scientific consensus could be achieved could contribute to a framework to guide policymakers and regulators on marketing and product claims pertaining to a healthy gut microbiome. Of course, thinking about what it would take in order to get us there, there's basic scientific agreement that if we want to understand both the benefits and safety from the standpoint of the framework, that there are several questions that we have to grapple with. From the standpoint of benefits, what is a healthy gut microbiome? How do we measure it? What is a beneficial physical effect? And does a beneficial effect matter in relation to health? And I think it's those connections across the continuum that are so critical to really moving this forward. From a safety standpoint, some of big questions also: what is an adverse effect with respect to the gut microbiome? Just because the microbiome changes doesn't necessarily mean that that's good or bad. And how do we measure these changes in relation to potential adverse effects as well as the beneficial effects? So despite the fact that we have these huge questions and are asking these, certainly gut microbiome science has been translated into a variety of products already. For those of us in the field, we spend a lot of time wondering, how did they get there from here, and really what are the underpinnings to a lot of the products that exist out there? I mean, this has been an issue in nutrition, not only in relation to the microbiome, but also the approaches using the genome similarly, and variation in genetics across individuals. So I think we've got a lot of work to do from a standpoint of understanding all of these pieces. So gut microbiome science certainly has been translated into a variety of products. I think we still ask, should it be? There have been a number of approaches to target the gut microbiome with regards to health effects and related to leanness, weight management, allergy, cognition, anxiety, stress. But again, a lot of those connections between effects on the microbiome, ultimate effects in relation to measurable health changes, particularly in humans, have been slow in coming. So I think where we are at the moment comes out of the history of microbiome committees within ILSI. In 2009, the Gut Microbiome and Health Committee was formed, and one of the first aspects of the work that was conducted there was to, again, try to define what a healthy microbiome was. This committee worked on this question for a couple of years. and came away with a number of conclusions, some of them which may sound vaguely familiar today still, that a healthy microbiome cannot be defined by a single idealized community composition. A healthy microbiome is more resistant and resilient to disruption. Certain microbiomes may increase susceptibility to infection and disease. However, it's still unknown if dysbiosis is a cause or consequence of the disease. And this was written up in a publication in 2012. And at the end of this work, the committee actually disbanded because it wasn't quite clear where ILSI was going to go to next in this regard, partly because the feeling was that the basic science wasn't there and we still had a lot of work to do before the application in the context of ILSI North America's interests and application was going to be lacking. That said, then moving forward a number of years, we get to 2016, and the recognition that this is still an area of great interest to ILSI. And so the committee was reformed in 2016 as the Gut Microbiome Committee, and so has been around for 2.5 years approximately, and has been working again to resurrect some of these questions and try to figure out where to go in relation to this area. And in December 2018, there was a workshop conducted in order to address some of these considerations again. And I'll talk a little bit about some of the findings that came out of this workshop, the objectives of which were to assess the state of the evidence on the gut microbiome and associated health benefits, determine if there's sufficient evidence to establish measurable gut microbiome characteristics that could serve as indicators of health, and identify short and long-term research needs to characterize a healthy gut microbiome, and to put this information out to the public, which this came out as a publication spearheaded by Mike McBurney in Journal of Nutrition this year. So some of the conclusions from this, and this really is then feeding into why we're here today, certainly some of the same things that came out of Backhead's publication in 2012. Microbiome communities are highly individualized, show a high degree of inter-individual variation to perturbation, as well as just the composition in general. And as we've learned over the past decade or so with more longitudinal data, tend to be relatively stable over years in healthy adults. There's a substantial functional redundancy in the microbiome, and I think this is one of the pieces where we are looking beyond just the characterization of microbial composition to think more about what is the microbiome doing, and some of the shortcomings in relation to the genetic approaches of studying the microbiome has missed some of the short and long-term dynamic changes that may be relevant to host health. Many of the particularly large-scale epidemiologic studies have often been limited to a single stool sample, for example, or if there have been interventions, they've been relatively short-term interventions. We've relied on the fact that the microbiome can change rapidly from a functional standpoint, and so using nutrition interventions that are short, where we may need to be thinking more along the lines of years to really see major shifts in the microbiome. And also the early reliance on 16S for sequencing and some of the limitations there. So some of the additional conclusions from the group was, we have yet to establish a mechanistic link of specific changes in gut microbiome with function, or specifically markers of human health. And this, for those of you who are, investigators who submit grants to NIH, you know that you can't nowadays just submit a grant and say, I want to see if I can change the microbiome. You really have to be able to show what is the relevance to a health outcome. So this is really, the health piece comes back again. And another aspect of this that really relates to our challenges in looking at causation versus associations is whether dysbiosis is a cause, consequence, or both of changes in gut epithelial function and disease risk. And so this led to a number of possible directions to go from here. I think one of the things that has become recognized broadly is that these are complex microbiome-host interactions, and it really requires a comprehensive multidisciplinary approach to this to bring the expertise to bear on the questions that need to be addressed. Biomarkers and or surrogate indicators of host function or pathologic processes based on the microbiome or relevant to the microbiome need to be identified and validated. And this has certainly been a challenge in research in relation to disease risk in a number of different areas. So this is not specific to the microbiome, but from the standpoint of we really need to have good, robust markers to be a part of this. And also, studies of exposure response that need to combine both validated microbiome-related biomarkers with the multi-omic characterization of the microbiomes to get more at some of the functional aspects of this. And sort of goes without saying that studies need to be designed and powered to account for variation, whether or not it's in the area we're discussing today or more broadly in nutrition research in general. So I think one of the things when we think about diet, gut microbiome, and health is, for those of us who come at this direction from nutrition, we've always had this, long-standing interest of what is the impact of diet on disease risk, particularly thinking about it in the context of chronic disease risk. However, you can put your favorite disease in here. And really the question has been, what is the impact of diet on these biomarkers? And when we think about the gut microbiome in this space, we can think about both from the standpoint of how diet affects the microbiome and then the potential direct effects of the microbiome on disease risk from the standpoint of direct effects of microbial components on the immune system or interaction with other receptor-based components in the epithelium. However, in relation to diet, I think our biggest focus and challenge here is really in the impact of the gut microbiome on producing compounds coming from the human diet that are then affecting the host. And so this is really the focus for today, to come up with some components that we can consider as monitoring aspects, particularly in relation to development of reference materials. So when we think about all the ways that we interrogate the gut microbial community, a lot of historically our work has been in who is there, with a focus on nucleotide measurements, both with regards to the 16S ribosomal RNA gene on the left, as well as shotgun sequencing of DNA for metagenomics. But really, where we think we're moving is more from the standpoint of the functional aspects, which are in the middle. What are the microbes doing? Certainly understanding better the expression of messenger RNA as well as proteins, and in the context of today's discussion, what are the metabolites or the downstream products of the functional aspects of the microbiome. So this is really a combination of the exposure to dietary constituents as well as the interaction with the microbiome. And so our overall objectives for today are to think about candidate metabolites with links to diet and health that could be used for quantification in a gut microbiome metabolic metabolomic reference material, and also to think about the challenges and consideration of these metabolites and their use and development in the reference material. And I think this is something that we will grapple with on many levels in today's discussions, but will really be the underpinnings of that. So with that, I will stop.